In this video, I want to discuss the steps in respiration, specifically the physiology of respiration. And there's going to be 5 main steps. The first one is ventilation, where you have to get air into your lungs. The second one, just to list them all out at the beginning, is external respiration. External respiration is once you have the air into your lungs, you've got to get it into the capillaries that are surrounding the alveoli of the lungs. So this is air in the lungs into the blood system. Then you've got to transport. So you've got to use red blood cells and hemoglobin to transport that oxygen out to the tissue. You then have to do something called internal respiration. And that is where you have to get the oxygen out of the blood and off the hemoglobin and into the tissues, specifically also into the cells, because in the cell you have to do cellular respiration, where the mitochondria are going to use that oxygen to make ATP, the cell's energy. This first video is just going to cover ventilation, and we're going to talk about the steps that affect ventilation. One of the first things I need to do is just get some lungs drawn up, and I am not particularly artistic. So there's my lungs. Try and draw some bronchioles. Give myself some alveoli. Obviously these are a lot bigger than they would actually be in the lungs, but just want to draw them out. So these are the tiny air sacs that the air is inhaled into. Another structure we need to get on here, I'm going to make it a similar color, is each of the lungs is surrounded by something called a pleura, and there's a very tight pleura that's right up next to the lungs. And this is called the visceral pleura. And each lung is surrounded by its own visceral pleura. So this would be the visceral pleura. There's an additional pleura, which I'm using similar colors because I don't know that they need to be separate colors, but just to give a little bit of a distinguishing color. This would be called the parietal pleura. And the parietal pleura covers each of the lungs together, so it covers them in one sac. The important thing about the pleura is it creates something called an intrapleural space. And that's going to be important as we talk about pressures and we talk about how you have what's called a transpleural pressure to keep the lungs inflated. So while we're talking about pressures, we should go through pressures quick. The first pressure that's important is atmospheric pressure. If I can back up, what we should talk about first are pressures. Atmospheric pressure is simply the pressure that's out in the atmosphere that's around the person. So obviously we would talk out here, but I'll just put it right here, that this is 760 millimeters of mercury is a typical atmospheric pressure. I guess I might give a little bit of color contrast here and use some different colors for different pressures. The next pressure that's important is something called intrapulmonary pressure. And this is the pressure in the alveoli. So that would be the pressure down in the alveoli. And to be honest, I wrote 760, but I probably should have wrote that it's usually around 759 to 760 to 761. So it will vary between those pressures. And in fact, if the intrapulmonary pressure is 759 millimeters of mercury, then the pressure inside of the alveolus will be lower than the atmospheric pressure, and so that will cause inhalation. If, on the other hand, the intrapulmonary pressure is 760 millimeters of mercury, then the pressure in the atmosphere, I should have said 761, then the pressure in the alveolus will be higher than the pressure in the atmosphere, and so this will cause exhalation. Let me say that again since I got a little mixed up. If the pressure in the alveolus is 759, then the pressure in the atmosphere is greater, so air will flow from the atmosphere in, and that will cause inhalation. If, on the other hand, we can get the pressure in the alveoli up to 761, then the pressure in the alveolus is higher than atmosphere, and that will cause air to flow out, and that will cause exhalation. Another pressure is this pressure in here in the intrapleural space, and so this is called intrapleural pressure. And that, again, is the pressure in the intrapleural space. And it's not exactly, but it's typically, and so we'll use roughly minus 4 millimeters of mercury compared to intrapulmonary. So if intrapulmonary is 759, then intrapleural would be 755 millimeters of mercury. If intrapulmonary was 761, just using the color coding, then intrapleural would be 757. So it usually tracks roughly 4 millimeters of mercury lower than intrapulmonary. Intrapleural is usually 4 millimeters of mercury below intrapulmonary. And that actually creates a suction that helps keep the lungs open. I suppose we can mention real quick why that suction is necessary. So the intrapleural pressure helps keep the lung inflated. Helps keep the lung inflated. Because there are two pressures that are trying to collapse the lung. One is the cohesiveness of water in the alveoli. So the alveoli are lined with water, and that water wants to form into drops, and that tends to cause the alveoli to want to collapse. The other force that makes the lung want to collapse is simply the elasticity of alveoli and thorax. So alveoli have these little rubber bands around them made by elastic fibers, and that's important for exhalation because when you breathe in, that's an active process, but breathing out is generally a passive process where these elastic fiber rubber bands around the alveoli help the alveolus collapse and force air out. You also have elasticity of your thorax, just your ribs don't necessarily want to be expanded, so they want to recoil as well. All of these things will make lungs want to collapse. So there needs to be a force to offset this lung collapse, keep the lung inflated, and that's intrapleural pressure. A term that's used to describe that is the transpulmonary pressure. And that is equal to intrapulmonary minus intrapleural. And again, that equals minus 4 millimeters of mercury. Transpulmonary pressure, if that's equal to 0, this would cause lung collapse. The term is pneumothorax. And you would either get air in this space or fluid. Equals lung collapse. The technical word for that is pneumothorax. Alright, so that describes the main pressures, but now we need to go into some other things, like something called Boyle's Law. Boyle's Law is going to help us to explain how these pressures are changed in the first place, because Boyle's Law says that pressure and volume are inversely related. So that means if you make the volume of a space go up, the pressure will go down. And if you make the volume of a space go down, then the pressure will go up. Maybe the easiest way to do this is to say that you've got a box with certain air molecules. And to be honest, where the pressure arises in these situations are when these molecules hit the wall of the box. That's where you get pressure. We talked about it when we talked about blood pressures, things in the blood hitting the wall of the vessel. Pressure arises when molecules in a container hit the wall of that container. Well, if you take that container and you make it smaller still, then things are going to hit the wall much more often. And so these molecules are going to hit the wall much more often. The way we can express that kind of simply is if we decrease volume, we increase pressure. Alternatively, if we make the box much bigger, well, now these molecules are not going to hit the wall as often. So if we increase volume, we decrease pressure. What this means for the lungs is in inspiration or inhalation, what we do is we increase the volume of alveoli and thorax, because according to Boyle's Law, increasing the volume will decrease the pressure. So now our pressure will go down to 759 millimeters of mercury, which means we will breathe in, or inhalation. In exhalation, if we decrease volume of alveoli and thorax, now we're going to increase pressure. So this gets us up to 760 millimeters of mercury. And this causes us to breathe out. Now the reason I left all this space over here is because the way we measure this is we graph or we track breathing. So we take a breath in and we can measure the volume of air that's moved when we take a breath in and breathe out and breathe in. So these are called pulmonary function tests. This would be a large breath in, a large breath out, and then continued breathing. So there's some key values here. This is an all-in value. So when you take a breath in and you breathe in all that you can, how much volume do you have in your lungs at that point? This value right here represents a normal in. This represents a normal out. So when you take a breath out, what's the volume of air that's in your lungs or that you were able to move? And then this is when you're asked to take an all out, breathe out all the air. Even after you breathe out all the air, there's always some air that's left, and this is called your residual volume. It's usually estimated to be about 1 liter. It can be calculated directly by breathing in helium, but usually it's just estimated. Now there's some values here that are important. Like there's something called an inspiratory reserve. And that's the difference between a normal in and an all in. So after you've taken a normal breath in, how much more air can you get into your lungs? There's also an inspiratory capacity. Which is after you've taken a normal breath out, then how much air can you get in? And these are values that relate to how well the alveoli can increase their volume to decrease the pressure to breathe in. Just a slightly different color. We'll do something called a vital capacity. A vital capacity is how much air you can move. So it's the difference between you've taken a deep breath, all the air in, and you've breathed out all the air. How much can you breathe? We have something else here that takes into account the residual volume, and this is the total lung capacity. And that's often abbreviated to TLC. That's the difference between all in, all the way down to the residual volume. So how much complete air can you get into the lung? And this is going to be a number that we're going to use later, or talk about later, to talk about how you can diagnose disorders of the lung. Specifically, how you can diagnose disorders which make it hard to get all the air into the alveoli or all of the air out of an alveolus. Some additional ones here. From a normal breath out to an all out, it's called an expiratory reserve. It's called the functional residual capacity when you're comparing from a normal breath out to the total air in the lungs. All right, so those are some pulmonary function tests, some spirometry things that will come up later, but I wanted to define some terms like inspiratory reserve, inspiratory capacity, expiratory reserve, functional residual capacity. Probably the two most important are vital capacity, and even more important is the total lung capacity. The next thing I want to talk about is something called Ohm's law. Now, most people that have heard about Ohm's law know that it generally is used to relate to electricity, but it really relates to anything where you're concerned about the flow of something, because the flow of that something is related to how badly something wants to move. In the case right here, it's the difference in air pressure. In electricity, it's the change in voltage, because voltage is what drives electrical current to flow. And that's always divided by resistance. Now, the reason to think about airflow into the lung as Ohm's law is it helps you understand there's two things that are going to affect this flow. Either your ability to change the pressure, and we mean the pressure change down here in the alveolus, or maybe there's a resistance that's in the way. So maybe the alveolus is working just fine, but there's some sort of obstruction up here that's getting in the way of the flow. And so there's two main different problems that can arise with lung disorders. The first one we'll do is we'll do the resistance ones. So there are resistance disorders. And these are disorders of the conducting zone. So we'll come back and circle this later, but these are going to be issues of the bronchi and the bronchioles. There's something wrong with the tubes that are getting air down into the alveoli. These are classified as obstructive disorders. So there's an obstruction that's preventing the air from flowing down into the alveolus. Some examples would be asthma, where there's a constriction of the bronchioles, emphysema, where the small bronchioles are usually destroyed, bronchitis, where the small airways, bronchitis, are usually blocked by mucus. And then there can also be things like tumors that obstruct the airway. Now the way you diagnose these, if we can come over here, is what you're specifically looking at now is this area of the curve. You're going to watch exhalations. Now it's plotted in a little bit of a different way. It's plotted kind of like this, where this is an all-in breath and this is an all-out breath. What you care about with resistance disorders is you care about how fast somebody can move the air out of their lungs, because if there's an obstruction, they may be able to get just as much air into their alveolus and just as much air out, but it's going to take longer because there's an obstruction. It's kind of like if you tried to breathe through a large straw, like a Starbucks straw. You'd still be able to get just as much air into your lungs and out, but it would take longer. So we need to look at how fast you can get the air out. One of the first values that you need is something called the forced vital capacity. And this is called the FVC. And this is how much total air can you move when you take a deep breath in, all the way in, and you breathe it all the way out. How much air can you move? That's called the forced vital capacity. So again, it's vital capacity, and it's called forced because you're going to do it under great effort. Another value that's important, if we put this at 0, we say this is 1 second, and this is 2 seconds. A normal person, like I've drawn here, should be able to get most of their air out in 1 second. An abnormal person, an abnormal situation, someone with asthma, the curve might look like this. So again, they still might get just as much air out. Their forced vital capacity may be the same, but it's going to take them a lot longer to do this. So now we need a value that says, well, how much air have you exhaled in the first second? And this is called the forced expiratory volume at 1 second. What's called the FEV1. So in a normal person, most of the air should be out in that first second. So technically the FEV1 over FVC will be greater than 70%. So you'll get greater than 70% of your air out in the first second. And somebody with asthma, the FEV1, how much air they can get out in the first second, divided by FVC, I guess in our case, it would be something like, boy, it looks like less than 50%. But the technical dividing line is 70%. Should have put a less than. It's less than 70%. And if you have somebody that you know that has an asthma attack, just ask them to breathe out really as fast as they can, and it'll be a much longer kind of exhalation. It'll take a lot longer for them to get the air out. So we look at resistance disorders by looking at FEV1 over FVC. You can also just look at FEV1 over a predicted FEV, so you can do a percent of predicted. And the predicted calculation is based on your height, your age, and your weight. So those are resistance disorders. And just to color code this a little bit, again, resistance disorders are going to be disorders essentially in the airways themselves. Make sure I don't cross over. And I did anyway. So resistance disorders are problems with the airways. The next thing we have are pressure disorders. So again, Ohm's law helps us understand that if we're interested in flow, there's two things that can happen. We can have a problem with resistance to flow, or we can have a problem not developing the pressure that causes the flow to happen in the first place, that causes the drive for the flow. And these are could be called respiratory zone disorders. So the conducting zone is basically the trachea and the large bronchioles, and the respiratory zone is where actual respiration occurs, and that's deep down in the alveolar ducts, the terminal bronchioles, and the alveoli themselves. These are called restrictive disorders, and that's a tough term because it sounds like resistance. Restrictive sounds like resistance. Resistance means there's a resistance to flow. Restrictive means there's something restricting the ability of the alveoli to change their size. And remember, the alveoli need to change their size because by changing their volume, that's how you change pressure. And by changing pressure, that's how you get inhalation and exhalation. Sometimes these are also called compliance disorders, because the alveoli are either not compliant enough in order to expand and accept airflow, so they can't increase their volume very well, or they cannot contract very well. They cannot decrease their volume. So some examples, and let's split these up into cannot increase volume. So if you have mucus in your lungs, you have mucus in your alveoli, mucus is sticky, so when it comes time for the alveolus to increase its volume, mucus is sticky, so it's harder to increase the volume. Fluid can do that. Lack of surfactants. Scar tissue, or another term for that would be fibrosis. You can also have a problem. We cannot decrease volume. That's specific to emphysema. In emphysema, the alveoli have lost their elastic fibers and they've popped into really large alveoli. So air will flow into the alveolus just fine, but when it comes time for exhalation and the alveolus has to kind of pop back into place because of those elastic fibers, it doesn't want to because it's lost those elastic fibers. So you cannot decrease the volume, which means you can't decrease the volume, which means you cannot increase the pressure, which means you can't breathe out. A good way to think about these is this sequence up here. These conditions right here make it hard to increase the volume of the alveolus because they're sticky and they make the alveolus hard to expand. If you can't increase the volume, then you can't decrease the pressure, and so these are going to cause difficulties breathing in. And where you'll see that is you'll see changes in their inspiratory volumes. With emphysema, the problem is the alveolus doesn't want to contract after inhalation because it's lost those elastic fibers, so you cannot decrease the volume, which means you cannot increase the pressure, which means you can't breathe out. And the disorders will be reflected here in changes in the expiratory volumes. We could draw that a little bit. We'll keep it somewhat simple. But we can say that if this is kind of going to just replicate this again, just so it's not so cluttery. If we say, and this is a normal spirometry, what we can actually expect in emphysema is they're going to not be able to get air out as well. So their volume curves are going to be much higher. And the major difference here is going to be they're going to have a huge functional reserve. I should say large functional reserve, because they can get air in, but they can't get it back out. Somebody with these disorders, to be honest, they may breathe normally. It might be a little bit smaller than normal. But when they go to take a deep breath, like they're going to go up the stairs, they cannot get as much air into their alveoli. And so where you're going to see a change in these individuals is largely in the inspiratory reserve. We'll put it here. So the inspiratory reserve is going to be smaller. We can also put one of these individuals just to see how this would look. This is the best way to do this, to look for resistance disorders is to look at FEV1. But if we were going to look at that here, maybe the way we'd see this one is all of the breaths would take longer. So when they go to take a deep breath, they might get the same volume, but it's going to take a lot longer. And so the orange would be resistance disorders or obstructive disorders. So just to highlight those over here, pressure disorders are problems with the alveoli. Now, one of the things you'll find is in almost every case, disorders are mixed. Now, I'm certainly no MD, and so I don't want to mislead that I know precisely what I'm talking about. But if you have mucus or something like that that's blocking the alveoli, it's probably also blocking the bronchioles. And so in that case, you're going to get a mixed disorder. So a mixed disorder let's just go ahead and put it here. A mixed disorder is going to encompass both the bronchioles and the alveoli, the small bronchioles. And usually the way this will be manifested is both TLC, so total lung capacity, and the FEV1 over FVC are affected. And just to get it on there, is most pulmonary disorders will be mixed. As both the small bronchioles and alveoli will be affected. So that does it for ventilation. We'll come back and do videos on external respiration, transport, internal respiration, and cellular respiration as well. Thank you. Okay, so in these respiratory physiology lectures, what I'm trying to do is take you through the steps in respiration. And step 1, or step A, was ventilation, which we covered in a video just recently. Now we'll go to external respiration. External respiration is once you have air into your lungs, what are the barriers, or what is keeping it from getting into the capillaries flowing through the lungs so that you can load up this oxygen in the blood or get rid of the CO2 out of the blood. Just for a reminder's sake, we'll keep listing the 5 steps. Transport is once the oxygen's in the blood and in the red blood cell and on the hemoglobin. Could there be any problems with the hemoglobin? Then there's internal respiration, which is once you've delivered this blood with its oxygen out to the tissue, are there any barriers getting it out of the capillaries, or could the capillaries be blocked or something like that? And the last step in respiration that we'll need to cover is cellular respiration. So this video here, we're going to talk about external respiration. With external respiration, there's really 3 things that we need to talk about. We need to talk about first, we need to talk about gas laws that relate to respiration. The second part of this is we need to talk about matching ventilation, which is air in the alveoli, to perfusion, which is blood in the capillaries of the lungs. And the third part of external respiration is either additional barriers in alveoli that prevent gas exchange. So the first one, we have to talk about a couple of laws really briefly. The first one is called Dalton's Law. And Dalton's Law simply says that gases diffuse independently of one another. So in our atmosphere right now, we kept talking about how atmospheric pressure is 760 millimeters of mercury. And so some of that for nitrogen is around 597 millimeters of mercury. So we're surrounded by a bunch of nitrogen. Partial pressure of oxygen, or the pressure of oxygen in our atmosphere, is about 159 millimeters of mercury. Partial pressure of H2O is 3.7 millimeters of mercury. Partial pressure of CO2 is 0.3. Probably the most important way that we're going to interpret Dalton's law as far as respiratory physiology goes is our O2 concentration. If the air is dry at 25 degrees centigrade, generally our partial pressure of oxygen is around 160 millimeters of mercury. But what Dalton's law tells us is if we start manipulating some of these other things, it could affect our O2. And so now if we say the air is humid at 25 degrees C, because we've upped our vapor or water vapor in the air, that means it's going to displace some of the oxygen. And so our partial pressure might drop to 155 millimeters of mercury. And then, on a hot, humid day, it always feels like it's harder to breathe. So if it's humid air at 37 degrees C, then the partial pressure of oxygen might be only 150 millimeters of mercury. So on a hot, humid day, there is less oxygen available to you, and that might affect your respiratory physiology. The other law is something called Henry's law, and Henry's law takes into account the various solubilities of the gases. So if we were looking at these concentrations in the air around us, I'm going to draw a really big nitrogen, indicating that there's a lot of nitrogen around us. There's a little bit less oxygen, and there's even less CO2. If we imagine that this was a glass of water, or if we imagine that this is blood, each of these gases have different solubilities. So nitrogen does not have a very high solubility in water or in blood. And so in your blood, nitrogen would actually be very low. Oxygen would be higher. And CO2 likes to dissolve in fluid. And so CO2 is much more concentrated in your blood than oxygen. And this is due to Henry's law. The thing about matching ventilation to perfusion, if you think about it, if you have air in your alveolus, you want blood to pick it up. And if you have blood there, then you want air to pick it up. So the lungs are very good at matching ventilation, air in the alveolus, with blood in the capillary. In fact, if you have a situation where you have an increase in oxygen in the alveolus and a decrease in CO2, then what will happen is the bronchiole, so the small airways, will constrict. And the reason they're constricting is because that alveolus has already got plenty of oxygen, and so you don't need to dilate the bronchiole in order to get more air in there. Lung capillaries will dilate. And the reason they will dilate is if there's lots of air in the alveolus, let's get blood into that alveolus so that that blood can pick up oxygen. Now, one thing that might help us to distinguish this is systemic capillaries. So lung capillaries and systemic capillaries are directly opposite. They work in opposition. Lung capillaries, if there's lots of oxygen, you need to get the blood moving because you want to get blood into that area, pick up oxygen, and out of there. Let's say you're working your biceps in a gym and there's low oxygen in that area, then what you want to do is you want to dilate to get blood. But if there's lots of oxygen in your left biceps because you're working the right biceps, then you want to constrict the capillary in your left biceps so more blood will flow to the right. So in tissue, if you have plenty of oxygen, constrict because you don't need blood flowing to that area. And maybe that's easier to see if we do the opposite case, which is if you have a low oxygen situation and a high CO2 situation. In the bronchioles, that means you don't have much oxygen in the alveolus that you'd like to have loaded with oxygen, and so you need to dilate the bronchiole and get air into that alveolus. The lung capillary, if there's low oxygen in that lung capillary, then we need to constrict, because we need to slow down blood flow, let the blood have more time to pick up that oxygen. Out in the systemic capillary, what you're going to want to do is dilate. So here, if the systemic capillary has low oxygen, it means you're working that tissue, and you need to dilate the capillary to get more blood flowing into there. It's actually a pretty important thing in lung function that this works, because if this is a lung, and these are the bronchioles, bronchi giving rise to the bronchioles. And we imagine that the arteries are bringing in blood too, so the pulmonary arteries are bringing in blood. Blood tends to want to follow gravity to the bottom of the lung. And so what matching ventilation and perfusion does is it allows the blood flow to the alveoli that actually have oxygen available to them. One of the things that exercise will do, if you're exercising, then the increased blood pressure from exercising will force blood into all areas of the lung. And so now you'll be able to have more area of the lung picking up oxygen. So in the case of no exercise, let's do it this way. In the case of no exercise, this is the only real part of the lung that's exchanging oxygen. There's not enough pressure to get air up into the top of the lung, and since there's no air up there, since there's no blood up there, there won't be any air up there. But when you exercise, the increase in pressure forces blood throughout the lung, and that's going to equal greater oxygen exchange. The major point being that you can move the blood around to find the air, so to speak. Now the last thing I want to discuss that has to relate to external respiration is barriers in the alveoli. And so let's just draw real quick some alveoli. I'm going to draw these as kind of intact alveoli because I want to demonstrate what happens in emphysema because that's one of the things that can affect external respiration is emphysema. One of the first things I just want to mention is what we're talking about here is we have oxygen in here. And what we need is for this oxygen to make it to a blood vessel. And we'll just extend it on past this one. So we have oxygen in our alveolus, and what we would like is to get that into the blood vessel. And that's essentially what we're talking about with external respiration, is once I get the air into the alveolus, can I get it into the blood vessel? There's three things that can create a barrier here. The first one is excessive fluid. So one, excessive fluid. So we're talking like pneumonia or bronchitis in alveolus is an additional barrier to oxygen reaching blood. And I always focus on oxygen, but obviously we're talking about exchange here. It's going to be harder for the oxygen to get out of the alveolus and CO2 to get out of the blood and into the alveolus. Another thing that can be a problem is mucus. And I should probably switch colors a little bit. So we'll go with mucus. Mucus in alveolus is an additional barrier Here, just to be less specific, but more inclusive, it's a barrier to gas exchange. So here, it's going to make it more difficult for this oxygen to make it into the blood. It's going to make it more difficult for the CO2 to get into the alveolus. The third problem will be loss of surface area due to emphysema. So what happens in emphysema is these would have been individual alveoli, and their structure is maintained by little elastic fibers. They basically provide some springiness, so some elastic recoil after you inhale that allows you to exhale, but it provides structure. And when these are destroyed in emphysema, small little alveoli pop into large alveoli. Now this significantly decreases the surface area available for gas exchange, because these oxygen molecules now are a long way away from a blood vessel. So there can be oxygen essentially in a dead space in the middle of this alveolus, where it's got to go a long way to make it to a blood vessel. So here, this oxygen only has to go a short little distance to get to a blood vessel. Short little distance to get to a blood vessel. Here, because we have a much larger alveolus, we have a lot further to go. And so there's a loss of surface area due to emphysema. Another way you could put that is there's increased dead space in the middle of large alveoli. So you've got these large alveoli. The oxygen in the middle is essentially in a dead space because it's so far away from the blood capillary. So excessive fluid would be disorders like we talked about in ventilation, where you have excessive fluid in the alveolus, so things like bronchitis or pneumonia. Excessive mucus would be anything that causes mucus to build up, so this would be like the flu or respiratory distress. And then loss of surface area is usually related to emphysema. So that takes us through external respiration. Thank you. So in these videos on respiratory physiology, I've been going through the steps in respiratory physiology, which involves ventilation first, which is to get air into the lungs. Be external respiration, which is once you have the air into the lungs, you need to get it into the capillaries, or you need to have gas exchange between CO2 in the capillary and oxygen in the alveolus. There needs to be gas exchange done in the alveolus between the capillary. Next is once you have gas exchange and you have oxygen loaded onto hemoglobin in your red blood cells, you need to transport it. Next is something called internal respiration, which is really dependent on the capillaries that are delivering gases to the tissue. So those capillaries need to be intact. And the last one is cellular respiration. In this video, we're going to cover transport. The first thing is just to point out, when we talked about Henry's law, we talked about how CO2 is more soluble than oxygen. So let's just talk about how oxygen is transported. So oxygen, I don't know why I made that a Q. Oxygen and CO2 transport. Got ahead of myself. I was supposed to say oxygen and CO2 transport. Mainly the way oxygen is transported is almost exclusively on hemoglobin, 98% on hemoglobin. And depending on your source, 1 to 2%, but obviously we're going with 2% in solution. So just dissolve directly into the plasma. Again, we said CO2 is more soluble, so it gets a little bit more complex here. About 23% on hemoglobin, which I'll abbreviate HB now. About 70% on something called the bicarbonate carbonic acid system. And we'll come back to that at the end when we talk about cellular respiration. And then it will definitely come back up when we talk about the urinary system, when we talk about acidosis, alkalosis. And so the rest of CO2 is dissolved in solution. Now I'll probably just put step 2 here, and I've got about 4 things I want to cover. But step 2 is how is oxygen carried on hemoglobin. It's usually best to kind of create kind of a graph. And I'm certainly no artist, but the way that I would typically draw this is hemoglobin is 4 separate proteins. And then it carries a heme group, which is an iron. And each of those heme groups can carry an oxygen. So the way it works out in total is you can have hemoglobin, but this would be technically called deoxyhemoglobin. Or, I'm trying to draw 5 sets of these. So what I'm trying to draw is, this is a hemoglobin molecule like I'm drawn here. Because what I want to point out is that hemoglobin can have no oxygens bound to it. It can have 1 oxygen bound to it. It can have 2 oxygens bound to it. It can have 3 oxygens bound to it, or it can be fully saturated. So these would be called saturated. These guys here called partially saturated. And then deoxy equals unsaturated. If you plot a graph where you put oxygen concentration in millimeters of mercury on the x-axis, and then how often the hemoglobin exists in this state versus this state, you'll get a plot that looks kind of like that. And it represents something called cooperativity, and the cooperativity involves that once 1 oxygen is bound, it's easier for the next one to bind, and once 2 are bound, it's easier for the next one and the next one. So it kind of speeds up its binding as you go along. Some critical numbers here is the concentration of oxygen out in the tissues is usually about 40 millimeters of mercury. And the concentration in the lungs can be upwards of 150 because that's atmospheric, but we'll just go with 100 millimeters of mercury. Now what this curve tells us is this is tissue. Should we write that? And this is lungs. Is when hemoglobin is leaving the lungs, it's got 4 oxygens on it. When it gets out to the tissue, so out to the big toe, and the partial pressure of oxygen is still 40 millimeters of mercury, we still have 3 oxygens bound to it. So what this tells us is that hemoglobin leaves lungs with 4 oxygen and returns with 3. So it leaves with 4 oxygen molecules and returns with 3. So general process, go out with 4, come back with 3. Go out with 4, come back with 3. So you really only drop off 1 oxygen. And those that work in a hospital setting and have done a pulse ox, they know this because pulse ox is, so that's the oxygen in that's the little red meter that you put on the end of the thumb and that measures the saturation of oxygen in the blood. And usually it's a greater than 92%. Anything less than that would usually be a sign if you're not oxygenating enough. And so that essentially means that you're almost always saturated with oxygen. And that creates a huge buffer. One of the ways that that buffer is realized is in CPR. You can cycle the blood several times and still deliver oxygen. So CPR has changed a lot, and it used to be that you had to give breaths I'm not up to date on my CPR to be honest, but you should have to give mouth to mouth. But if you're unwilling to give mouth to mouth, you should still perform CPR, because just circulating the blood is still circulating oxygen. It's still circulating hemoglobin that has oxygen. So you might circulate it out and you drop off 1 oxygen. Keep it circulating and you drop it to 2. Keep it circulating and you drop it to 1. So there's a huge buffer on hemoglobin of oxygen availability. Speaking of hemoglobin, it's very, very genius, because hemoglobin drops off oxygen in high temperature, high acid, presence of something called DPG, which is a metabolite, and high CO2. Now, the way I like to draw this is essentially you want hemoglobin to drop off oxygen when there's work. When you're working a biceps in the gym, you would like the hemoglobin that's passing through the biceps to drop off oxygen. So when you're working your biceps in a gym, which admittedly doesn't happen very often for me, there's an increase in temp in that biceps muscle because it's being worked. What that does is it decreases HB's affinity for oxygen. So if you warm up hemoglobin, it doesn't like oxygen as much. What that means is O2 will be dropped off. So you warm up hemoglobin, it doesn't like oxygen as much, so the oxygen is kicked off. That means O2 will nourish the tissue. That is doing work. If you work a tissue, you will cause an increase in acid. So you'll be undergoing metabolism. Acid levels will increase. Acid levels will make hemoglobin not like oxygen as much. So hemoglobin will drop off oxygen, and the O2 will be available to nourish the tissue. If tissue is being used, then it's going to burn ATP. So using ATP will increase a metabolite called DPG. DPG makes hemoglobin not like oxygen as much. So hemoglobin affinity goes down, oxygen is dropped off, O2 will nourish the tissue. The last one is an increase in CO2 will also do this. And this is called the Haldane effect. Essentially what it says is CO2 and oxygen compete for hemoglobin. So if there's more CO2 around, it will kick the oxygen off the hemoglobin. That's good because now the hemoglobin is available to nourish that tissue. Now when that hemoglobin gets back to the lung and there's an increased concentration of oxygen, now the oxygen will knock the CO2 off the hemoglobin so that you'll breathe it out. So it works out pretty well that CO2 and oxygen compete for hemoglobin. Out in the tissue where CO2 is at a higher concentration, CO2 will kick oxygen off of hemoglobin and jump on the hemoglobin and be taken back to the lungs where now the oxygen concentration will be higher. So the oxygen will kick the CO2 off and the oxygen will be loaded onto the hemoglobin. The last thing that seems mildly appropriate, even though we often cover this in blood as well, is just to talk about the anemias real quickly, just to remind people of the anemias. Anemia is caused by, I'll just split it off right here. It's either not enough blood, and these are typical causes of this are hemorrhage. So that's hemorrhagic anemia. You can also have hemolytic. And so this is blood loss. Hemolytic, RBCs destroyed. So either there's sepsis or there's something destroying the red blood cell. Sickle cell, I guess, would fit into that too. And then aplastic anemia is bone marrow damage, We'll just call it marrow damage. You can also get anemia because of not enough hemoglobin. And this is typically iron deficiency. No iron to put into the hemoglobin, so you can't make the hemoglobin, or pernicious anemia. Pernicious anemia is lack of B12. B12 is important for making methionine, and methionine is the first amino acid in all of your proteins. So if you don't have B12 and you can't make methionine, then you can't make protein in general. One of the first proteins that's going to be affected is hemoglobin. You can also get a B12 deficiency because your stomach needs to make something called intrinsic factor that protects B12 through the digestive process or as it's passing through the stomach. So essentially a stomach disorder can also lead to pernicious anemia because you don't make that protective factor called intrinsic factor. The other main type of hemoglobin are abnormal hemoglobins. And this is sickle cell. Sickle cell is going to be interesting as we talk about internal respiration, and also it feeds into external respiration, because what is normally a nice round red cell becomes long and sickly. The hemoglobin actually pops together into these large strands, and it pushes the red blood cell into kind of a sickle shape, and that won't pass through a capillary very well, so it will block capillaries, leading to problems with internal respiration and external respiration as well, because both of these involve capillary exchange. And this one is, the last one we'll do is called thalassemia, which is, again, it's another mutation in the hemoglobin molecule that makes it difficult for hemoglobin to carry oxygen. So that's transport. So let's start this one more time like we have been, which is we're going through the steps in respiratory physiology. The first step is ventilation. You've got to get air into the lungs. Second one is external respiration. Once the air is into the lungs, you need to get it into the blood. That's called external respiration. Once it's in the blood, you've got to transport it, so you have to have proper red blood cells and hemoglobin in order to be able to transport it. Then you have to do internal respiration, which is once you have the oxygen and the gas in the blood, you need to be able to get it back out. And the last thing is once you get it back out and into the cells have to know what to do with it, which is called cellular respiration. And that's a process where mitochondria use that oxygen to make ATP. Now these are going to be fairly short, and so I'm going to do these together. Do internal respiration, cellular respiration together. We'll do internal over here. And the only thing I really want to point out with internal respiration is it's highly dependent on capillaries. This requires intact capillaries. Because what we're talking about is, I suppose the way we could do this is, if this is the lung and this is a blood vessel, this is external respiration. And I want to clarify that because we think of external respiration as breathing out. This is supposed to be a blood vessel. Looks kind of like a zombie finger. Internal respiration is, okay, we've got this oxygen into this blood vessel. Now I need to get it into the cells. So these are supposed to be cells. So this again is a blood vessel. I realize with my lack of artistic ability, I should probably just label better. So this is internal respiration. So just to clarify that internal respiration is not breathing in and external respiration is not breathing out. External respiration is let's get the air from the lung into the blood. Internal respiration is once it's in the blood, let's get it out to the cells. Now, one thing I suppose that's probably worth noting is the direction of transport is generally in this direction because there's more oxygen in the lungs than there is in the blood. And similarly, oxygen will go in this direction. CO2 that's Maggie. She's a little hyper. Good dog, but hyper. And CO2 will tend to go in this direction. So I had a scavenger hunt come to my house, so I've been finding paper clips and things like that. So I don't know exactly where I was, but I think probably what I was talking about is the forces that drive oxygen into the blood are that there's a greater concentration of oxygen here and a lesser concentration here. There's a greater concentration of CO2 in the blood that drives it out and into the lungs. Same thing happens in internal respiration. One reason this may be relevant to the Z states is if somebody is breathing in a bunch of CO2 because of smoke inhalation or because of smoking, then that will drive CO2 back into the blood. Or rather, it will prevent CO2 from leaving. The same thing can happen here, is if you deliver this CO2 because it loaded here, then you're going to be delivering CO2 to the tissue, which means it's going to be hard to offload CO2. So things like smoke inhalation will deliver CO2 to the tissue. Or probably more accurately, or will prevent the ability to rid CO2. To get rid of CO2. Another thing is, what if that blood vessel, if it's a capillary, and so it's very, very small, and again, we're trying to deliver blood to cells. What if we have a cyclic cell in here? So a sickle cell anemia. So sickle cell anemia will decrease internal respiration. It's probably likely too that it doesn't necessarily need to be a sickle cell to do this. You can probably get the same effect if you have high blood glucose. So if you've got a bunch of glucose as in diabetes or high blood glucose, will probably also cause the blood to be sticky blood. And this will also, so diabetes may also impair internal respiration due to increased blood thickness. As sugar is sticky. So internal respiration, it's offloading oxygen from the capillary into the cells or picking up CO2. There was a little bit random here, but tried to show a couple of different pathologies that can affect that. So if you've got smoke inhalation, then you're loading up CO2, which means you're going to take CO2 to the cells, which means you won't be able to pick up CO2. And so those cells might become acidic because of that excess CO2. Another condition, blood glucose. If there's increased blood glucose in the blood, it's going to make the blood sticky. It's going to be harder for the blood to flow, and that may impair internal respiration, especially at the capillaries, because that's where the stickiness is going to be most apparent. Also, sickle cell anemia, the red blood cells can turn into a sickled shape. Red blood cells pass through capillaries one at a time, essentially. And so if you throw in a sickled cell like that, it can completely block that capillary. That capillary won't then be able to deliver oxygen to these cells, so that will also impair internal respiration. Our last condition is cellular respiration. And my apologies if you are hoping for an in-depth run through the Krebs cycle and glycolysis. I'm going to keep it really, really simple. In fact, I'm not even going to necessarily put the numbers on it. Glucose plus oxygen is going to yield CO2 plus H2O plus ATP. And again, really in a layman's term, what's happening here in cellular respiration is this is a mitochondria, and mitochondria has these cristae are folds. And where you get your ATP is you stack up hydrogen molecules on the inside. You know, I may be drawing this wrong. Got ahead of myself. You stack up this hydrogen on the outside. And what you put on the inside is this oxygen. My apologies, started drawing too fast. So you put all this oxygen on the inside. And what happens is this hydrogen does not like to be stacked up on the outside. One, it wants to diffuse on its own. Two, it's attracted to that oxygen because if it can get to that oxygen, it's going to make water. And so the net result here is this hydrogen will go to this oxygen, and the result will be the CO2 plus hydrogen plus water plus ATP. So you let this hydrogen go to the oxygen, you'll make water, and you'll make ATP. And the carbons that are left over from the glucose will be attached to the oxygen as well, and that'll make CO2. There's some disorders that can affect this, like Lay's disease is a disorder where there's a slight impairment of the ability to make ATP. I mean, if you have a major impairment of this process, you're never going to be in the first place. But if you have a slight impairment, some of the results will be going to have difficulties with the tissue that uses the most ATP, which essentially would be neurons. Now, before I end here, I just want to talk one last time real quick about, as a preview of when we get to the urinary system, the connection between CO2 and acid and oxygen. So hypoventilation is essentially decreased breathing rates. And what this can cause, I'm putting arrows here because I want to make sure that this arrow just only goes one direction. Hypoventilation means you're not breathing enough, and that can cause a buildup of CO2. And if CO2 builds up, CO2 in the body can be essentially thought of as acid. And so that's going to decrease your pH. A decrease in pH is the same as an increase in acid. And this is also going to cause a decrease in your base, which in our body is usually bicarbonate, HCO3. This is going to cause acidosis. Now, one of the things that you can do to cure acidosis, since it's an increase in CO2, is simply increase breathing. Another term for that would be hyperventilation. Running out of space. Now, the reason these arrows are one way is because hypoventilation will lead to an increased CO2, increased acid, decreased pH, decreased base, and acidosis. And the corrective mechanism is then to cause an increase in breathing or hyperventilation. Now, these things over here, anything that causes hypoventilation and leads to an increase in CO2 is called a respiratory acidosis. But you may get in this acidosis state because your base is dropped or because you've consumed too much acid in your food or something like that. So you can get in the same situation. Anything out here can also cause acidosis. But if it's not caused by respiratory, then this is called metabolic acidosis. Now, the reason I'm doing this is just to start to preview how the respiratory system is involved in acidosis. It's also involved in alkalosis because in hyperventilation, and this would be an increased rate in breathing, and I don't mean like rapid shallow breathing, I mean rapid deep breathing, because if you rapidly breathe, what happens is you blow off your CO2. And CO2 again is essentially acid, so if CO2 is going down, acid is going down. If acid is going down, that means you have an increase in pH. That's just the way the pH scale works. Is a decrease in acid means a high pH. And this means a high base or a high HCO3. And this is called alkalosis. Cases of alkalosis, as long as the respiratory system is not causing it, The respiratory system will try and cure alkalosis by breathing slower. You'll breathe slower. Or this will lead to hypoventilation. You can have disorders that cause you to hyperventilate. They're usually psychoneuroses or things like that, or drug overdoses and drugs, things like that cause you to breathe fast, and that gets rid of your CO2. This is called respiratory alkalosis. But not all cases of alkalosis are caused by the respiratory system. You can have other systems like, for example, you usually absorb bicarbonate from your food, and so if your food stays in your digestive system longer because you have something like constipation, then you'll absorb more base. And so now the problem is not respiration, the problem is that you're absorbing too much base, and this would be a cause of metabolic alkalosis. So we're going to talk about this much more in depth when we actually get to acidosis and alkalosis, but I want to at least give a preview at this point so you're starting to see the connection between the respiratory system, CO2, and acidosis and alkalosis. Thank you.